like pigs was easy to obtain, right? Sexual maturity only about 5 to 6 months. And the mice cannot mimic human disease, such as cystic fibrosis, as I mentioned. And also, like rodents, like mice and humans, that have huge difference in metabolism and hemodynamics between mice and humans. But pigs are more similar from the side of the physiological and anatomy, metabolism, pathology, right, quite similar. And also, mammary gland cells correctly translate post-transcriptional vitamin K dependent proteins during hemostasis. So, and also for size considerations, right? So, like rodent blood flow, you are very difficult to measure, right? But for pig, it's quite easy. And also for genomic considerations, pigs can perform genetic modifications of genome, and also genomics are quite close to humans. And also, as a preclinical animal model can be accepted, since pigs are now primates, so they can prevent the spread of viruses. Since like two clones share the same viruses, such as like HIV, as from monkeys, primates to humans. So pig is acceptable for preclinical animal model. So far, there's a lot of pig models was created. So here's a list. I think if you go to the PubMed to search for it, you can always find more and more. So here give you few examples. First is pig models was created after the CRISPR-Cas9 technology. So it's this one. This is the one that's VWD. That is like a patient for what? That's a the syndrome. The syndrome, like both man and woman can got this disease, right? This genetically is autosomal dominant. So typical symptoms is prolonged bleeding time, right? And decreased adhesion or platelets to glass beads, reduced or no aggregation of resistin aggregation. Anyway, just and all like plasma 7-factor related antigen or clotting activity decreased or VWF activity was decreased. Yeah, that's a typical symptoms. So once the CRISPR-Cas9 technology was developed, so the scientists in the CAS, that is also Chinese work, they quickly did this, create this animal model. So this is for CRISPR-Cas9. They did 5 experiments, finally got 3 knockout pigs. So they did the phenotypic analysis. It's pretty easy. You just look at their antigen, VWF antigen activity, right? And to look at clotting, like factor 8, this factor 8 activity, and also bleeding time. That's a more very basic phenotype that we can measure. So as expected, this antigen activity, the factor 8 activity was significantly decreased in the homologous pigs. That means 2 allele was added, right? Knockout pigs. And bleeding time was significantly increased. So this paper was published on Cell Research 2014. That's just like 2 years this CRISPR-Cas technology was created. very quick following work. So second example was the Huntington disease pig model. The Huntington disease is like dense disease, like neuronal degenerative disease, right? So that's a rare autosomal dominant disease. Patients generally develop from middle age and have motor, cognitive, and mental symptoms. So okay, I will not go through these details, but have the motor neuron. That means like we walk is affected, right? Cognitive, mental symptoms. So that is in West prevalence of Western people is 4 to 8 in 100,000 people. It's a rare disease. Okay, so again, they found the disease-causing gene is HTT, the Huntington HTT, which encodes HTT protein. So the scientists in CAS, they published this paper on Cell that they, last correspondence author was scientists who made the salmon fish oil people. He was the first author for that paper. But after studying in the United States, he came back to China, and right now in Guangzhou CAS research. So he already published a lot of good paper, talked about made a lot of biomedical pig models. So they used the CRISPR-Cas technique. They made the Huntington disease pig model, and they say there are a lot of defects, right? They have defects, and especially they monitored the pig's walk style. It's not that regularly as compared to the wild type pig. So these pigs have the lower body weight and the survival rate, like 30 years, about 3 years old, before 3 years old, about 80% of these modified pigs die. Yeah, so it's mimic the human Huntington disease very well. So let's provide animal model for further study this pathology of Huntington disease. Also, this pig model can try clinical trial. You can try some drugs, right? So that's the reason why this paper can go to Cell. It's quite a good study. Okay, that's a biomedical models. The third one is the memory bioreactor. That means we can do transgenic pigs, right? From the mammary gland, we can produce proteins that from any other strategy we can only get few amount of the protein, but with the mammary gland we can have more. So here is the good paper talked about engineering protein processing of the mammary gland to produce abundant hemophilia B therapy in pig milk. That's also from the University of Missouri, Dr. Brothers lab. So here is give their summary is only with 900 transgenic pigs that will supply all of these proteins worldwide. Yeah. So this is their study. Mammary gland bioreactor. Another we have to talk about is xenotransplantation. So here's we can say lot of people on the waiting list for the organ. Right?
Kidney transplantation.
Yeah, waiting for transplantation. So the donor is quite few, and a lot of on the waiting list. So with the scientists' hard work, we can say the xenotransplantation already make a lot of progress. From the pre-1980s, the organ, like patient, like the monkeys, the experimental animals can only survive for a few minutes, right? So right now we can like months to years, like monkeys, can survive with a pig's heart for several months or years. So right now scientists work together to make the more progress, like permanent, right? So that's a lot of works here. So the pig is generally considered to be the most suitable xenograft source animal because of its size, favorable bleeding characteristics, and the similarity of many of its organ systems to those of humans. Okay, this availability, safety, I will not go through that. Quite similar with why we use pigs as animal models, right? Size, breeding characteristic. Oh, this is quite important since pigs have larger size. Why not consider sheep and cattle since they are like only one babies, right? But pigs, the litter size you can have more. It's easy to harvest, easy to get these animals. So here is the quite a similar strategy. For example, we also the fibroblast. We do the gene edit. We can take embryonic fibroblast have nucleus. Like pig have the endogenous retrovirus in pig. Yeah, have like a 50,
endogenous.
Yeah, 56 or 58. The pig have their own retrovirus in their body, but this virus was not harmful for pigs.
Pig.
Yeah, this is in pigs.
Inside
but
the
everything
DNA?
is fine.
Inside the DNA?
Yeah, inside the DNA. So, but this is a threat for the
Humans.
organ transplant, right? Even there's no harm for in pig. Nobody can say it's no harm for humans. So.
Laushi, is the same mechanism like bacteria include like in the CRISPR discovery?
Oh, that's a good question.
Is the same evolutionary?
Yeah, I don't know for sure, but I just know there's a retrovirus about 56 or 58 retrovirus
So I think
in the pig, but they are no harmful. They just exist over there, but no harmful pig.
But it is possible that during transplantation, maybe.
Oh yeah. So,
It is possible.
yeah, after transplantation,
Media is changed
maybe
in humans.
it will be harmful to humans. So that's so previous people have no idea how to deal with this retrovirus, right? But here CRISPR-Cas, this technology easily to knock down all of them. So knock down all of them, and we can have this
Free.
organs, to go to the human. So that's also like do transgenic, do a nuclear transfer. So people always think about go to the human body, to decrease the hyper injection.
Yeah.
Right? So still have a lot of genes can cause this acute hyperinjection.
Rejection.
So there's a lot of genes that was modified, and this is for xenotransplantation to avoid the hyperacute rejection. Animal models was created. Okay, so it's what's going on. Here is the first one, GGTA1 knockout mice. So this is this person is also the first author this paper. So GGTA1, I think I mentioned the last class. So knock out this gene to avoid the hyperacute injection. So, but this pig, this knockout pig, was allowed to go to the market for people which was allergy with the meat. Okay, so this is already marketed genetic pigs, genetic modified pigs. So, but the major purpose for this knockout pig, GGTA1 knockout pig, was for xenotransplantation. Okay, so GGTA1 knockout pig transplant heart to the hemidrias, avoid hyperacute rejection. Then this monkey lasted for 179 days. You know, hemidrias. I don't know how to pronounce it very well.
Okay.
Hemidrias. Okay, and this is the Nature Medicine 2005. Okay, that's the big person, right? I mentioned for the members resurrection. So this George Church talked about, okay, the human face beast heart is coming soon. So what did he do? His group just knocked out all 56 or 58, I didn't remember correctly, you know, like the retrovirus in pigs. So they call this pig 1.0, pig additive 1. So that's just to suppress, knock out all of the retrovirus in pigs. And pig 2.0, let me see, they have pig 1. It's a man who got the first pig heart transplant. So named David. David got the first pig heart, but he lived for died after 2 months.
After 2 months?
2 months, 2 months.
Oh.
So here is the 2 months.
सुअर की दिल लगा के जल्दी मर गया.
Maryland Medical Center, David genetically modified pig's heart, passed away on March 8th. So lived for 2 months following surgery. But the doctor say, you know, it's not just like a heart failure. It's just died from other reasons, not heart failure. So this is the first brave person, David. And here is the second pig heart. So this, Lawrence just, you know, after surgery, lived about 6 weeks. 6 weeks. So they are both of them are very brave. Actually, you know, think about, okay, if I have a pig heart, I'm so scary, right?
I also.
So right now, you know, pig is so, you know, good animals. We talk about pig taking wings with a genetic modification. So a lot of work, scientists work with pig for xenotransplantation right now in China also. Gene editing in pigs. What else? That is also growing human organs in animals. This is for this part. That's our team. We can also do targeted gene editing and by the nuclear transfer to make the genome genetically modified animals. That's outstanding.